my career progression has kind of mirrored that as Luminex's development. When I started out my career in the early 2000s at MGH, one of the tasks I was given was going through our cryobank of a bunch of old serum and plasma samples left over from clinical trials that we had ongoing. And we wanted to see what other information we could glean from those samples. And that included just one, seeing if there was enough sample that we could actually perform any other colorimetric assays with. And if so, what assays could we actually perform? And also, being early in my career, I was motivated a lot by free lunches. And one day, Bio-Rad had a lunch and learn where they were really talking about their new multiplex kits that they were starting to offer. So after attending one of these and seeing what we could actually look at with such a small sample set, I went and talked to my PI, and we tried to set up a demo where we actually could pretty much buy the largest multiplex that they offered at the time to run a lot of these precious samples. And we were really amazed with the data we could actually glean from these samples, especially since a lot of these trials had already ended. We had a hypothesis about what was going on with those patients over the times of these treatments. And by running these multiplex, instead of just having one cytokine that you're looking at, or maybe two, you could actually look at a whole panel. So you could actually see the onset of CRS or all sorts of other things that we had hypothesized were happening without actually knowing. And then being able to correlate this with the flow data was very powerful for us. And it also allowed us to redefine the way that we actually looked at our sample analysis. These patients are bled repeatedly. And by being able to maximize the use of these samples, it could actually limit, and we could actually reduce some of the blood draws that we needed for some of these patients. And it was actually very meaningful to us to actually try and make their lives a little bit more comfortable. And we were just really pleased with what we could learn from such a small sample set. And then as I went on to industry in the late 2000s, I still primarily thought of Luminex as a cytokine screening tool. And it wasn't until later that we started really using it for epitope mapping when we were working with alternative scaffolds and trying to tailor binding accordingly to what we were trying to do. And then using some of the early work with the QuantiGeneplex, they also were very, very useful for us. And it's kind of come full circle for me as I got into cell and gene therapy, where I currently work now. And I'll just kind of give a little brief background on what we do. So the current standard of care for AML patients is a matched healthy donor will be mobilized to push stem cells out of the bone marrow into the periphery where we can collect them with apheresis. Following identification of a patient, the patient will then be myeloablated, and then we will actually perform the stem cell transplant. After the stem cells have had time to engraft, a lot of times there's a targeted therapy that is also used for these patients. However, one of the big problems is without having any sort of editing done to these cells, a lot of the stem cells and the AML cells co-express a lot of the same targets, leading to, again, myeloablation of the patient and on-target bystander killing, essentially, thus leading to possible loss of the graft. So with VOR, we are actually kind of a middleman in here. So we'll take the healthy donor HSCs, we will edit them to knock out and make a target null HSC that is then implanted into the patient where it has time to engraft. And this way, targeted therapies can also be used on these patients without the bystander killing that would happen to your graft, thus kind of making them invisible bone marrow cells and leading to healthy engraftment. And most of these edits that we are doing, there are known null in the general population, so there's not any sort of worry about health crisis there. And this is really important, especially for patients with AML that typically have a refractory period and a relapse within 1 to 3 years of transplantation. And this is the highest unmet need and one of the areas that we would like to really change with our treatment therapies. So Without the VOR approach, many of these targeted therapies fail within, or they don't even get to market due to just myelosuppression and other health concerns and toxicity concerns. With the VOR approach, we can actually make these target null HSCs, we can do the transplants, we can actually start to treat and use some of these therapies that have actually been sidelined due to these health concerns and use on these patients, giving them a whole new opportunity and hopefully delaying MRD. And so this really opens the door for treatment of these people so we can actually have a true complete remission instead of a complete remission followed by onset of a secondary disease. Right now, we have a very simple and elegant process where there's no new genetic material utilized in our edits or viral vectors. There's no cell expansion. We essentially get the starting material. We enrich the stem cells. We are currently using CRISPR-Cas9 and electroporation for editing. And then it is formulated, cryopreserved, and ready for the patient. Our total production time is 3 days, and our target vein-to-vein time is 7 to 10 days. And this is critical for these patients. They do not have a long time to wait for a matched donor or a graft. So we're really trying to keep this process quick and efficient as possible. And we all know cell and gene therapy is complex. I just presented about the simplest case you can have for cell and gene therapy. You have the nucleic acids, the DNA, the RNA, the nucleases that you're going to use, either Cas9, Hi-Fi, whatever, lentivirus, AAV, the growth factors that are going to be utilized in maintenance of your cells and health of the cultures. All of these will have to be evaluated either in process samples or in the final drug product for residual screening. Therefore, it's important that we have an instrument that is capable of working in multiple matrices that can have either a protein or a molecular readout. And another thing that I can't stress enough, and why I posed the last question about the GMP compliance, is if you can actually develop your assay on an instrument that is also GMP compliant in research, you can continue to improve that assay all the way through tech ops and down to manufacturing. So it's really important, in our opinion, to actually be able to do this and streamline as much as possible. So at the last minute, you're not having to do all sorts of bridging studies and changing your methodology for your release assays. And luckily for us, the legacy FlexMap 3D has been able to really fit the bill for us. And as we had already developed a lot of custom assays to look at free Cas9 and the growth factors associated with the culture of the product, we started to think about the RNP complex. This is the actual heart and soul of the editing. So it's where the guide RNA complexes with DNA to hone to the area on the gene that we'd like to cut and perform the double-stranded break. However, we typically use an excess of guide RNA to ensure that there's enough in the system to complex with the Cas9 to make an efficient cut. And as we were thinking about this, we wanted to know if we could actually use the Luminex for this purpose. So guide RNA is a 100 nucleotide single strand RNA. It's got a 20 nucleotide variable region followed by an 80 nucleotide common backbone. There's also a high degree of secondary structure in this backbone. And as we were contemplating how we would do this, if we could do this with the Luminex, we were thinking about how would we want to target this? Do we want a universal target that would only bind the backbone? However, as the industry moves to more multiplex or multiple edits, we didn't really think this would be very useful because it would just say, yes, there's guide. We want to know exactly which guide can we detect. We also wanted to explore if we could actually do a full length probe. This would be used for, one, detecting the guides, discriminating them, but we also wanted to know if we use this, could we start to use it for identity confirmation? Could we use it to look at truncations in the guides? Because different vendors provide different quality of guides, and that's one of the things that we've always looked at is the quality of those guides, how many of them are actually full length, the structure that you're actually paying for, that percentage. And then as we started thinking about the variable region, this would actually be the most useful for us and hopefully the easiest to develop. At the time we were using digital PCR, I guess we still are using digital PCR for this. It is RNA isolation followed by reverse transcription to make the cDNA, and then we'll droplet generation and read the plate. And as we thought more about this, I was thinking of the microRNA workflow from the cookbook. And it's one of the reasons I really love the Luminex cookbook, how you really encourage us to think about what can be done with the instrument, and why meetings like this, I believe, are so important is it lets us get together, throw ideas around, and see what works. And sometimes they do, sometimes they don't, but this one actually worked pretty well for us. The microRNA is about 24 nucleotides, around the same length as the variable region of the guide RNA. While digital PCR requires more reverse transcription and amplification, the Luminex would be more driven by probe specificity, hybridization efficiency, and the RNase digestion, which we really thought would be more equivalent to what would actually be in the drug product itself, and that's what we really care about. We all know that PCR can definitely be skewed in different amplification steps, and especially in our reverse transcription as well. And this really brought us to our collaboration with the LuminexPLORE Lab. At the time, VOR was a really small company. I think there were about 25 of us. We were in a very small lab that we were outgrowing, and we were in the process of moving. And this was about March of 2020, so the pandemic had just hit. We were in the process of building out a new lab, and we wanted to get the Luminex in before then, but one, we were out of space in our lab, and we kept getting delays from the shutdown of construction and everything else. So for us, it was kind of a no-brainer to really work with Tim and Jackie to actually get this set up. And we really had clear objectives. We wanted Luminex to work with the external vendor to design the probes and run the proof of concept study, optimize the assay with hopes of being able to transfer it back to VOR after we moved into our new lab and had the Luminex set up there. So we were able to provide Luminex with cells and guide RNA, and they went through several different iterations of trying multiple guides. We did learn that full length is really difficult to do for an RNA single-stranded guide capture, which we weren't too surprised by that. But other than that, things went really well. They were able to spike the guide RNA into the cells, lyse the cells, extract the RNA, do the RNase digestion, add the streptavidin PE, and run on the Luminex. They were able to generate multi-log standard curves showing that this was possible. And probe 1 and probe 2 were the same sequence. The only difference is a 5-carbon spacer between the chimeric X-tag DNA portion of the probe and the complement RNA sequence. So we were really pleased with this and won the sensitivity of it, and I can't imagine what we'd be able to do with the IntelliFlex if that time comes for us. We were able to transfer this assay back into our shop, where Yanju was able to take the same standard curve that she had used for qualification of the digital PCR assay, just run it on the Luminex, and we got very nice standard curves as well. And it really recapitulated what we saw with the digital PCR. We had good recovery, a little bit of variations there, but all in all, for an unoptimized assay that we just brought back in and just ran once, we were really pleased with this. And in comparison to the digital PCR method, which takes about 7 hours for the RNA prep, the extraction, RT-PCR, digital droplet generation, read the plate, and data analysis, it still takes a full day, and it uses several different instruments to actually do this. While the Luminex, we're able to actually do the same gRNA prep and the extraction steps However, the RNA prep and amplification, the plate hybridization, all this happens in the thermocycler, which really makes it really easy because they're using essentially one instrument for doing most of these steps, followed by the wash, reading the plate, and data analysis. It takes about 5.5 hours, but it's something that we'd probably be able to start to pare down as we started to optimize this. Another thing that we really liked is there's a lot less room for technical issues to occur. We all know that RT-PCR and digital droplet generation is not always as smooth as we'd like, and there are issues. And also, even after you generate the droplets, you still need to read within a small window of time. This is another thing that we can avoid by using Luminex and the direct hybridization. So we're really pleased with our collaboration with Luminex and that it was capable of detecting and quantifying the guide RNA. We did have a faster readout with less hands-on time, which will hopefully lead to less operator fatigue and room for error, and also less PCR bias, since we are actually just finding what's actually in the drug product itself without any amplification or artificial means of skewing that data. They were prompt, maintained timelines. We received regular status updates, and it was extremely smooth tech transfer back into our shop. And after this, we really started to think about what else we could use Luminex for. All the same assays that we had used to develop for residuals in our drug product and in process samples, we'll also be using and modifying to use as identity assays for everything going into the manufacturing suites. And this will include guide RNA, nucleases, viruses, growth factors. Everything that will be used for the manufacturer will also be probably screened on a Luminex if possible for identity confirmation. Another thing that we are also really starting to evaluate is editing confirmation. While we can do a lot of this with NGS, NGS is extremely expensive, time-consuming, and would not be fun to qualify that assay. We are also using Sanger-based methods, but again, you have to then have a license to usually utilize those tools, and even going into a commercial setting, that is not something that we'd like to do. By using Luminex to actually, after research, has really identified the indel profile that we expect to see in our drug product, we can actually start to utilize Luminex with probes for those indels and identify them. So it might not always be for a complete release, but it could be for a conditional release while we're still waiting for the final data to come back from these other orthogonal methods. And as we get into, or the industry moves toward base editors and other editing, multi-editing in a single cell, we really believe that Luminex has a very strong place there. As we look at even base editings where you're looking at just a corrected single nucleotide sequence, if you are performing multiples edits in a single cell, Luminex's ability to have multiple beads, look at all those targets at once, it could be a very powerful tool for us and a much quicker readout and something cheaper and easier than NGS. We could also use it for off-target assessment and other known translocations that could occur depending on our editing strategies. And it really just comes down to cost savings in the end. Anytime you can have one instrument that can do so many different things, but it does them well. It doesn't do them halfway. Luminex has been sensitive. It takes up less bench space than a lot of other instruments. And since we are starting to develop a lot of these assays in research and use them all the way through our manufacturing, we believe it's a very good investment. And I'd like to thank Heather, Steve, Jackie, and Chad for actually performing the work at Luminex, as well as Yinju, Napul, Brittany, and Eric at VOR, who really put a lot of the work in to actually perform all these assays and get things really going on the utilizing this for a more molecular basis in our lab and getting away from just our typical cytokine screens. So with that, any questions?